Hi, my name is Shawn, Jayee. And in my project, I was finding out whether MNR1 leads to the metabolic flexibility in ovarian cancer cells.
Speak high. Speak high.
So when I was doing my project, my problem was finding out. My problem was patients diagnosed with ovarian cancer have a 30% chance of survival after 5 years. The reason for this is ovarian cancer has an aggressive metastasis and it goes to metastasis early and is characterized by micrometastasis. Micrometastasis will then lead to occlusion, which then leads to the patient's mortality. This all takes place in the perineal cavity, and the metastasis is easily spread to the GI tract and the omentum. So my general objective was understanding the mechanisms that promote ovarian cancer metastasis. So part of my hypothesis was the metabolic flexibility of ovarian cancer cells is required for the metastatic potential. So based on previous research in the lab, I was able to come up with a more specific hypothesis that MNR1 is a primary regulator for metabolic flexibility of ovarian cancer cells. Metabolic flexibility is when a cancer cell has a stronger mitochondria and is able to survive against inner and outer stresses that normal cells would not usually be able to survive against. So what is MNR1? MNR1 is a protein that regulates genes such as itself and other genes. It also causes anti-apoptosis and improves the mitochondrial respiration or induces ATP production. Depending on the location of the MNR1, the cellular location of it, the proteins it's bound to, it causes the anti-apoptosis or the improvement of the respiration. So my specific objectives. My specific objectives was to first find where MNR1 is located in different ovarian cancer cells. This could be in the nucleus or the mitochondria or even both. And then the second objective was to find what other proteins MNR1 bound to in the different ovarian cancer cells. Does binding to these certain proteins increase the metabolic flexibility of these cancer cells? So the first method, in order to find the first question of where MNR1 is found, I need to use a process called fractionation. And fractionation is a process used to separate the cellular components of the cell. And because the cells are lysed, it separates the proteins that are contained in the nucleus and the proteins that are contained in the cytoplasm, or more specifically, the mitochondria. So in order to do fractionation, I need to do a BCA to get the protein concentration before running the Western blot, and a Western blot to find if MNR1 is found in the nucleus and or the mitochondria of different ovarian cancer cells. The cell lines I used for fractionation was the TKO cell line, the OVCA 432 cell line, R182 cell line, and the F2 cell line. So these are the results. In the TKO cell line, you can see that the MNR1 is located mainly in the cytoplasmic fraction, but I also had to use an integrity control and a loading control when I did the fractionation. An integrity control is to see whether all of the fraction in the cytoplasm is really all cytoplasmic proteins, or did I get some of the cytoplasmic proteins in the nuclear fraction. NDUFS3 is a gene located in the mitochondria, so it should only be in the cytoplasmic fraction of the fractionation. However, there's some contamination, and some of the NDUFS3 appeared in the nuclear fraction of the fractionation. ILF3 is a gene located in the nucleus, and so it should only be present in the nuclear fraction of the fractionation. And as you can see, there's an enrichment of ILF3 in the nuclear portion, and there's a little bit of contamination in the cytoplasmic portion. And GAPDH is used as a loading control to make sure that both proteins were equally concentrated and put in the fractionation. In the F2 cell line, you can see that most of the MNR1 is located in the cytoplasmic portion, and there's a little bit of MNR1 located in the nuclear portion. However, in both the ILF3 and the NDUFS3, there's a contamination of both of them. So based on the conclusion of the first of where it's located in the TKO cell line, majority of the MNR1 is present in the cytoplasmic or the mitochondrial fraction of the fractionation. And the F2 cell line, I cannot conclude whether MNR1 is in the cytoplasmic or the nuclear fraction because of the contamination located in the nuclear and the cytoplasmic fraction. So in order to find what proteins MNR1 is bound to, I used a process called immunoprecipitation. Immunoprecipitation is where there's a set of beads and I put the lysate on the set of beads and the non-specific proteins bind to the set of beads and I take out the lysate, and the lysate has now specific proteins, and when taken out, I labeled that the supernatant. The supernatant has all the proteins that I'm looking for, and all the non-specific proteins that bound to the bead were thrown away. And then next, a new set of beads were put, and an antibody for the proteins I'm looking for was put in the set of beads. And the lysate was also put on, and the beads bound to the antibodies, which bound to the proteins I was looking for. The lysate, which now didn't contain the proteins that I was looking for, was taken out and labeled the wash. And the set of beads now contain the antibody and the proteins I was looking for. And then once the set of beads were taken out, I was left with the protein I was looking for. The reason I use this is because MNR1, if it was bound to the proteins I'm looking for, which I'll explain later, which are COX4 and BCLXL, when Western blotting them, I would be able to see both the BCLXL and the MNR1 because they would be bound together, or the COX4 and the MNR1 because they're bound together. So in order to do this, I lysed the proteins, which broke down the cells, and then captured proteins for analyzation. Then I did a BCA to get the protein concentration, and then I immunoprecipitated to isolate MNR1 and what is bound to in different ovarian cancer cells, and then I Western blotted it. However, when isolating the MNR1, because there is no antibody for it, I really isolated the BCLXL and the COX4 first to see if it was bound to the MNR1. So what is BCLXL and COX4? So when MNR1 is bound to BCLXL, which is another protein, it causes anti-apoptosis. And when MNR1 is bound to COX4, it induces cellular respiration. So the cell lines I used for this were the TKO cell line, the MR182 cell line, and the F2 cell line. So this is the immunoprecipitation, and as you can see, the IgG is used as a control, and then the COX4 is the data I'm looking for. So as you can see, there's COX4 present in the input, which shows that there's COX4 in the F2 cell line, and then there's also COX4 in the IP. However, in the IP, there's no MNR1, showing that when I found the COX4, there was no MNR1 bound to it. So this is the BCLXL IP. As you can see, there's BCLXL in the input, and there's BCLXL in the IP. However, there's no MNR1 in the IP, meaning there's no BCLXL and MNR1 bound together. In the TKO cell line, you can see that there's COX4 in the input, and there's also a little bit of COX4 in the IP. However, there's once again no MNR1 in the IP. But for BCLXL, there's a lot of BCLXL in the IP, and there's some MNR1 in the IP also, showing that when I immunoprecipitated for BCLXL, MNR1, because it was bound to the BCLXL, came with it, and in the Western blot, both were present. So what I can conclude from this is in the TKO cell line, MNR1 is shown to be bound to BCLXL, and the F2 cell line, the results are inconclusive. So in my general conclusion, in TKO, most of the MNR1 is found in the mitochondria, and most of the MNR1 bound is shown to be bound to BCLXL. In F2, MNR1 is present in both fractions, but it's not clear where a majority is located because of the contamination. And because of this, the results could not demonstrate whether it binds to COX4 or BCLXL. And that's the end of my presentation. I would like to thank Hussein and Dr. Alvaro for helping me out in my project, and Alyssa for doing all the organizing it all. Thank you.
Any question? No questions? No suggestions? Very good, my friend. Thank you.
Thank you.
Our next presenter is Sanjana Kandiraju from International Academy Central.